Good afternoon, everybody who is joining. Welcome to this Psychiatry virtual seminar. While we wait people joining, I'd like to start the session. So I'm Hiro Morishita, Associate Professor of Psychiatry in this department. Usually Dr. Baxbaum hosts this meeting. He has a schedule conflict, so today I'm excited to host on behalf of him. So we're excited to have Dr. Jessica Abels here today. I think she's a familiar face to all of us. In many ways, Dr. Abels started her lab as assistant professor 2 years ago, 2019. She originally got MD PhD training at UT Southwestern, but then she completed her MD training at Mount Sinai. Then she took a unique path. So instead of directly going to psychiatry residency, she took postdoc at Rockefeller with Nat Hines and Ines. And there she worked on nicotine addiction using a rodent model. And some of you may know her seminal work studying nicotinic alpha-5 receptors, key role in habenula pathway. She also had started some exciting, quite innovative work on NO gas, how that impact addiction. And after the postdoc, she come back to Mount Sinai, started her research track residency at Sinai. Not only she started her clinical training, but you may know her as a chief of research residency, really pushing research side simultaneously. And she worked with Paul Kenny neuroscience department to continue her nicotine addiction study. So a lot of really exciting work she accumulated, but then as a faculty, she really started open up really innovative work connecting brain and body, which is a new exciting direction of research. So today you will hear her new lines of research about the effect of hyperglycemia on reward circuitry. So thanks, Jess, for joining. Please take it over.
Thank you, Hiro, for that nice introduction. Give me a moment to share my screen.
Just one note, we should have a lot of discussion time at the end. So if you have question, put in your comment or Q&A session. And also we will discuss that at the end. And if you can raise your hand, I'm happy to open up for question too. All right, thank you, Jess.
All right, can everyone see my screen okay?
Yep.
Okay, perfect. All right, so again, thank you everyone for giving me the opportunity to talk to you guys today. I'm really excited to share our project looking at how chronic hyperglycemia, the hallmark of diabetes, affects reward circuitry. And just briefly, I have no disclosures. All right, so diabetes is a common and increasing illness, both here in the United States and worldwide. And I think it's particularly appropriate in this 100th anniversary of the discovery of insulin to really take a look at where we are in treating diabetes. Certainly, we've made large advances in improving the lifespan of diabetes, but along with that, we have found that we're not doing a good job of preventing the development of diabetes or the consequences that come from living a lifetime with the disease. So just some figures quickly to point out. Nearly 500 million adults worldwide have diabetes. And this is actually rising. So this is approximately 1 in 11 people, and it's estimated to reach more than 600 million people by the end of this decade. And diabetes is associated with an increase in blood glucose, And this uncontrolled glucose leads to several complications, including cardiovascular disease and effects on both the peripheral and the central nervous system, including blindness and neuropathy. Not surprisingly, given the effects on the peripheral nervous system, diabetes is also associated with effects on the central nervous system. And in particularly, I'm interested in the incidence of mental illness among those patients with diabetes. So some very seminal studies done in the early 2000s showed that there is almost double the rate of major depression among diabetic patients. So around 21% in patients with type 1 diabetes who have a deficit in insulin production, but even higher, almost 30%, so 27% among those patients that have type 2 diabetes or insulin resistance. So basically their insulin receptor is not functional. Hyperglycemia itself, or elevated blood sugar, has been associated with states of dysphoria, fatigue, and anger, all characteristics of depression. An A1C, or a measure of glycated hemoglobin, a long-term measure of glycemic status, correlates with depression scores. So you can see that in the graph here on the right, where as the hemoglobin A1C increases, the probability of having a clinically significant depression on this particular scale increases. And just for your reference, diabetes is defined as a hemoglobin A1C above 6. So you can see really it plateaus at an A1C above 8, but you can see that there is a nice correlation here between the level of glycemic control and depression scores. Additionally, diabetes is associated with increased anxiety, both generalized anxiety as well as anxiety that's specific to the disease. And work from James Murrow's group has actually shown that the presence of diabetes is also a risk factor for suicide. So all of these pieces of clinical evidence really point to the fact that there does seem to be an effect of diabetes on reward circuitry. Some other clinical evidence also supports this conclusion. For example, in neuropsychological assessment of patients with diabetes, they're found to have impaired executive function, largely in processing speed and cognitive flexibility and attention. But also other studies have demonstrated that there are alterations in the gray matter volume in areas within the reward circuitry in both type 1 and type 2 diabetes. While many of these areas are overlapping, there are some specific effects of each disease. So even though they both share hyperglycemia as their common denominator, the mechanisms are different and do appear to have different effects on the brain. And interestingly, type 2 appears to have more severe effects than type 1. Animal models also of diabetes have also provided some evidence that there's long-term effects of hyperglycemia on reward circuitry. Specifically, monoamines levels are altered in diabetic animals. Similar to the cognitive impairments in patients, they also demonstrate impairments on memory tasks such as the Morris-Water maze. As diabetes progresses, animals also demonstrate impaired sensory motor gating on PPI and behavioral despair on forced swim or tail suspension tests. At baseline in normal animals, insulin activates cholinergic interneurons in the nucleus accumbens to regulate dopamine release and signal reward. However, there's been mixed results in the preclinical animal models of diabetes as to what this actually means for reward processing and motivation behaviors. And so we'll explore that a little bit later in the talk. So despite all of this evidence that diabetes really does affect the brain, I found it quite shocking that there's really a lack of an understanding of the effects of diabetes at the molecular level. So in here, looking at gene expression or transcriptional profiling, I actually am hard pressed to find many papers in the literature looking at how diabetes affects gene expression in the brain. And I highlight these two papers here to give you an idea of how recently this field has really started to emerge. So this first paper here is from Ronald Kahn's group, and notice that this was published just in August of this year. And this is looking at how insulin regulates gene expression broadly in the brain in intact animals. So we're really, this research is really in its infancy. This paper here from Yuxin Yan's group, published in 2019, is really the first example I could find of post-mortem analysis of gene expression in diabetic patients. So interestingly, both of these papers point to some of the same brain areas that have been highlighted in the imaging studies from diabetic patients as well, with insulin regulating genes mostly in the hypothalamus, which is not surprising given that that is where most of the metabolic control is thought to occur, but also in the hippocampus and the nucleus accumbens. So type 2 diabetes seems to also affect gene expression in some of these areas, but interestingly more so in the caudate nucleus and then the hippocampus, so consistent with those cognitive deficits, but then more limbic structures such as the amygdala and the accumbens, as well as the anterior cingulate. So again, both of these two studies are pointing to the nucleus accumbens and suggesting that there is a direct effect of hyperglycemia on reward circuitry. And so that really leads us to the two main questions that we're exploring in the lab currently. So what are the effects of chronic hyperglycemia on gene expression and key reward areas? And then next, how does this affect reward circuitry function and stress susceptibility? Trying to kind of connect those dots between what we see molecularly and epidemiologically. So the approach that my lab takes is a multi-omic approach to profile the reward circuitry under hyperglycemia. We're utilizing a mouse called the NuTrap mouse that allows us to do simultaneous profiling of both the epigenome and the translatome in a cell type specific manner. So generally what we do is induce hyperglycemia in these NuTrap mice, and we wait either 6 or 12 weeks for chronic hyperglycemia. And then we take different areas of the brain reward circuitry. So either the nucleus accumbens or the striatum, a brain area called the habenula, or the midbrain, including the VTA. And then afterwards, we will send those for profiling. This is very broad and general and not cell type specific yet. We'll get to the cell type specific in just a minute. I would also like to give you a little background as well about the two types of broad models that we will use. So again, as I had mentioned, diabetes comes in two flavors. There's a type 1 flavor, which is insulin deficient, and a type 2 flavor, which is thought to be largely due to insulin resistance or inefficient signaling through the insulin receptor. There are several models for each of these types of diabetes in mice, but we'll largely be using the streptozocin model, which is a beta cell toxin that you administer to the mice to prevent them from making any more insulin, or a diet-induced obesity model, sometimes used in combination with a small amount of STC. But just so you're aware, there are other models of diabetes available as well. So we've done the most work with the STZ model, and I would say it is in general a pretty good model for inducing diabetes, but it does have some limitations that we should consider. First, the model itself is somewhat variable. So the efficiency of beta cell ablation is sensitive to the time of day that you administer STZ. It's also strain dependent, and we found that we, even within our own lab, have different results with different people injecting the STZ. Additionally, as you can see here, it is sexually dimorphic. So these are blood glucose levels in male and female mice administered various doses of STZ. And you can see the dose range here. And again, you can see that not all of the animals at the lower doses become hyperglycemic, and that there is a much larger response in the males than in the females. And this is consistent with what's in the literature, but it does make it somewhat challenging to use the females in this model. We've also found that we can measure A1C in these animals, and even though the females are not as profoundly hyperglycemic, there is a significant increase in their hemoglobin A1C after STZ administration. But again, I'd like you to notice that in the females, this is below the threshold for diabetes. So going forward, I'm going to show you data primarily from the males in this model. And again, this was work that Christy Nivlo and Zainab Okitokun in my lab have worked together to generate. All right, so back to the question. So the reward circuitry is quite large, and where do we start and which cell type should we start with? There's no shortage of choices to choose from, as well among the neurons, as well as astrocytes and other glia. We could start in the accumbens, we could start in the VTA, but I've actually chosen to start in the medial habenula for a few reasons, and I'll discuss with you that next. So the habenular circuitry is, it is part of the reward circuitry. For those of you who study the reward circuitry, you might not be as familiar with this area, but it is kind of the hub between the limbic forebrain signals and then the feedback from the mid and the hindbrain. You can see that the habenula sits here really at the intersection between signals coming in from the forebrain and integrating and funneling them down to the midbrain to provide that feedback for the output from the midbrain back to the forebrain. So this is one of the reasons we chose this area. Another reason we chose this area, I'm sorry, I had left my note in there for myself, is that these cholinergic cells appear to be quite vulnerable. So the habenula here, shown in green, is largely comprised of cholinergic neurons. And you can see their cell bodies sit here in the medial habenula and project down to the midbrain. And here you can see their terminals in the midbrain. Cholinergic neurons are also important in the nucleus accumbens, where they're interneurons that regulate the dopamine release. But in this particular area, we've decided to start looking at the cholinergic neurons because other studies have shown that loss of these cholinergic habenular neurons leads to anhedonia, so a symptom of depression. Additionally, cholinergic neurons have to balance their energy demands with neurotransmitter synthesis. So acetylcholine is a direct product of the electron transport chain Krebs cycle. And so the animals really must balance their acetylcholine or their acetyl-CoA needs for either energy or neurotransmitter synthesis. This then leaves these neurons vulnerable to metabolic disruption. And we can see that from clinical studies looking demonstrating reduced viability of cholinergic neurons in other brain areas, so in the septum or the hippocampus of both diabetic patients and patients with Alzheimer's disease. For those of you who had attended Grand Rounds recently, you may have noticed, remember as well, that cholinergic neurons in the forebrain are upregulate chat as an early sign of stress in Alzheimer's disease. And this was work done by Dr. Muffson's group. So based on these reasons, we thought to start here looking at these cholinergic neurons in diabetes. Additionally, this happens to be the area that I've worked in as well for nicotine addiction. So I'm quite familiar with profiling this area. So our first pass was really to kind of follow up on what we had seen or what Dr. Muffson's group had seen as well, and just basically look at the levels of chat expression itself, or the enzyme that makes acetylcholine, the rate-limiting enzyme for acetylcholine synthesis. So we thought we would look at the expression of acetylcholine in these diabetic animals after 6 weeks of hyperglycemia. And what we found is that if you look at the different areas of this projection, so either the cell bodies, the projection fasciculus retroflexus, which are the axons, or the terminals themselves, we could measure the chat levels. And we found that similar to the early signs of stress in mild cognitive impairment and early Alzheimer's disease, that levels of chat appear to be slightly elevated after 6 weeks of hyperglycemia. Not quite significant, this is still somewhat preliminary data, but particularly the levels seem to be elevated in the axons and the IPN. So again, suggested that there is some stress induced in these neurons by hyperglycemia. All right, so what about at the molecular level? So here again, just to remind you, we're using a chat neutrap mouse and inducing diabetes with STZ and examining them 6 weeks later. And we're looking only here at the cholinergic cells in the medial habenula. And the way we're able to get this cell type specificity is by immunoprecipitating these GFP tagged ribosomes, specifically from these chat neurons. For your reference, we are pooling about 5 animals per sample, so there's not a lot of material that we use. And again, this is a preliminary data set, so not complete yet, but still able to reveal some interesting results. So based on the blood glucose, we can divide our STZ treated group into hyperglycemic or normoglycemic. And we can compare that to a vehicle-treated control. And when we look in the hyperglycemic group, we see that there are 176 genes that are upregulated by hyperglycemia and roughly 1,000 that are downregulated. And this is work that's been done by Zainab to generate the tissue, and then with the assistance of RT from Li Shen's group to analyze the data. So when we look at the genes that are up or down regulated, we did some pathway analysis to try and see if there are any patterns that pop out to see what perhaps hyperglycemia is doing to the function of these neurons. And what we find for genes that are upregulated in hyperglycemia, we see genes such as insulin biosynthesis and processing. And this is somewhat reassuring in this model where we have a deficiency in insulin. So this does again suggest that these cells are trying to respond in some way to this deficit in insulin. But we also see pathways consistent with upregulation of neurotransmission, both of nicotine, and this is a norepinephrine driven pathway here, as well as Wnt signaling. And this is consistent largely with that upregulation of monoamines that we had seen in the patient populations as well as in the animal models. When we look at the genes that are downregulated in hyperglycemia, it's actually somewhat surprising at first that it's largely clotting pathways, so prothrombin and fibrin complement coagulation cascades. However, this again is consistent with the idea that the cholinergic neurons are under an amount of trophic stress, and is consistent with the idea that there is reduced NGF processing, which requires prothrombin, and is similar to what Dr. Mufson had observed in his early Alzheimer's disease cases and the cholinergic neurons in the forebrain there. So even though this is a very preliminary data set, the data that we're able to get from this is consistent with what's been observed in the literature. So our conclusions for the first part of the talk is that these habenular neurons do demonstrate some signs of stress as measured by the increased chat fluorescence. We are seeing genes associated with neurotransmission upregulated and genes associated with NGF processing downregulated. All of these three things together suggesting that these cholinergic neurons appear to be uniquely stressed with hyperglycemia. So our future studies really are to focus on completing this initial data set, as well as optimizing our co-IP protocol so that we can IP both the nuclei and the ribosomes from these same tissues to do that simultaneous epigenomic profiling at the same time. And then beyond that, what we'd like to do really is extend our studies to more well-known areas of the reward circuitry, and as well as more well-known cell types within the circuitry, such as dopaminergic cells and glutamatergic GABAergic cells. But we're also interested in looking at astrocytes, and I'll touch on that a little bit more in the next part. And currently we're also starting as well studies with the diet-induced obesity model so that we can profile more of a type 2 diabetes data set. All right, so that kind of wraps up the first part of the talk and our first question at how diabetes affects gene expression in the reward circuitry. But as a psychiatrist, I'm somewhat more interested in how chronic hyperglycemia actually affects the reward circuitry functionally, and perhaps how it interacts with stress to increase affective disorders. Much of what the work that has been done in animal models and diabetes has largely been very basic characterization of diabetes, of diabetic animals, using tasks such as open field or light dark to assess anxiety behaviors, and forced swim or tail suspension tests to assess depressive behaviors. But for those of you who are basic neuroscientists, you'll know that the field is really trying to move beyond these basic measures because they don't really capture the kinds of deficits that we observe in clinical populations. And there's been a real problem trying to translate preclinical findings into successful clinical treatments. And so we have done some basic characterization, and similar to the reported literature, we find that in tasks such as open field and light dark, we do see even somewhat modest results at baseline in these diabetic animals, and even conflicting. So here within our own results, the STZ animals spend less time in the center, which many would interpret as being more anxious. But on the other task, on light dark, there's no significant difference between the two groups. And again, this is not a significant result. So I would say that this is a somewhat modest and contradictory effect at baseline. So our approach is really to move past these very simple tasks. And so what we're starting to do is to use more complicated and clinically relevant tasks largely operant tasks with the help of Zainab and Roman from the Russo lab to assess reward processing. So either using a task that Roman has developed, the probabilistic reward task, or using tasks that really assess rewards, such as condition place preference for natural and drug rewards. As well, we're starting a project with Brian Swice to really look at how hyperglycemia affects decision-making, flexibility, and reward processing on a task that he's developed called Restaurant Row. And so we feel that these, and additionally, I should say that this probabilistic reward task and the Restaurant Row task have also been done in human patients. So we feel that these are really much more clinically relevant behavioral assessments that we will start to begin. What I'll show you right now is a little bit of operant task that we have done kind of as preliminary data before progressing on to the Restaurant Row task. So what I'm showing you here is some operant learning in STZ treated mice. So these are STZ treated males, again, 6 weeks after that initiation of hyperglycemia. And what we're asking these mice to do here is to lever press for a food reward. So we fast the mice first to 85% of their baseline body weight, and then put them in a box with 2 levers and train them to press one lever for a food pellet while the other lever gives no reward. And initially we train them on a very easy ratio. So this is called a fixed ratio 1, meaning that 1 lever press yields 1 reward. And despite the literature demonstrating learning deficits in hyperglycemic mice, you can see here that our hyperglycemic mice in red actually performed much better on this task, even on the first day. So performing twice as well as the normal glycemic controls. The hyperglycemic controls, you'll see there's no data here, largely because they didn't need the training. So we spent a little bit more time with the controls trying to get them up to speed. So we gave them an extra day of training on the FR1 ratio, and you can see their performance did not improve. So what we did then is to fast the control mice a little bit more to make them perhaps a bit more motivated for the food. And what you'll see then, now the control mice are fasted to 85% or to 80% of their baseline weight, while the hyperglycemic animals remain at their 85% of baseline. And you'll see with the increase in fast, the control animals' performance improved. Now this pattern of improved performance by the hyperglycemic animals persisted as we made the task more difficult. So here you'll see that on a fixed ratio 3, meaning that now it takes 3 lever presses to get that food reward, the hyperglycemic animals are still outperforming their control counterparts. And again, the hyperglycemic animals were able to proceed on to an FR5 task, whereas the normal glycemic controls did not. So this to us, there are several reasons why this might have occurred. So diabetes is well known to induce hyperphagia, or decreased sensation of satiety. So these animals do eat more. So, perhaps that's reflected then in more hunger and more motivation so that they perform better on this task. Fortunately, we had trained them up to press the lever so that we could specifically assess their motivation for food using a progressive ratio task. And what progressive ratio does is it asks the animal to press more and more for each successive reward. So the first reward may require 1 lever press, while the next reward requires 2, then 4, and exponentially increases, so on. And eventually, the animals reach a point at which they will give up, and we call that the break point. And that is usually considered an assessment of their motivation for that reward. So interestingly, despite outperforming their control counterparts, there actually was no difference in the progressive ratio breakpoint between these two groups. So indicating that there actually is no difference in motivation for food between the two groups. So somewhat contradictory, even within our own data, but also interesting and something that allows us, we can kind of parse these different aspects apart in the restaurant road task, where we get a much richer data set and can ask more challenging questions. All right, so that was kind of our baseline behavior, looking at just general reward processing. And we see that, you know, in our hands, as well as in the literature, the animal models really are demonstrating a modest depression or anxiety profile at baseline. And our own operant data is inconsistent with an impaired appetitive drive, despite the fact that clinical populations, it's very clear that diabetic patients have increased rates of depression and anxiety. So are we missing something here? Is a two-hit hypothesis, or is a second hit needed to kind of recapitulate these increased incidence of mood disorders in the clinical populations? And so the next part of the talk will really focus on the intersection of stress and diabetes as risk for developing depression. So our first pass was to see if subthreshold stress would affect anxiety behaviors in diabetic animals. Here we have a nice cohort, this time of both male and female STZ treated mice. And for whatever reason, this particular group of females responded quite well to the STZ. So we have nicely hyperglycemic males and females in this group. And so what we did here is, again, we waited 6 weeks after induction of hyperglycemia. And then we subjected them to a very mild stress. In this case, restraint stress for 1 hour on 2 consecutive days. And then we assessed their levels of anxiety in the light dart task. And what you can see here is that there is a baseline difference between males and females and their level of anxiety. But there's an even further decrease in both males and females in the hyperglycemic group. So while overall, the post hoc did not survive significance here between the control and hyperglycemic by sex, overall there was an effect of stress on the hyperglycemic animals, such that they were more susceptible, in this case, spending less time in the light after this very mild stress compared to their control counterparts. So our next research focus will really try to use this subthreshold stress to see if we can induce more of a depressive phenotype in the diabetic animals. And we're going to use 2 different types of subthreshold stress, micro defeat or mild unpredictable stress. And again, these are done in collaboration with Roman from the Russo lab and Samantha and Mohammed in my lab, who have just joined. Additionally, we'd like to look at the animal's response to antidepressants after these subthreshold stresses, as the literature really suggests that diabetic patients with depression are harder to treat and tend to be more treatment resistant. So our goal is really to see, are there certain antidepressants that are certain classes that are more effective in diabetic patients? This would really improve the clinical treatment in this population. All right. that's left over, apologies. Some of our future studies as well kind of come out of some follow-up studies and trying to look at the blood sugar in females and try and understand why the females are more resistant to developing hyperglycemia. And this has been observed as well in patient populations, that there is a protective effect of estrogen. However, we know that pregnancy is a time when females are more susceptible to blood sugar dysregulation. And we have observed this as well by taking some of those STZ treated females that were resistant to developing hyperglycemia, and then seeing whether or not pregnancy would change their hyperglycemia later. This also then gives us an opportunity to study the transgenerational effects of maternal hyperglycemia on the offspring, which Sam is really interested in doing. And here is looking at anxiety in a marble burying task in those F1 hyperglycemic pups. Mohammed is also very interested in looking at the mitochondrial function and morphology in neurons in the hyperglycemic brain, as well as neuron astrocyte coupling within the reward circuitry, to focus again on other pathways that might be dysregulated in the hyperglycemic brain. And that really concludes most of what I have to present today. So a lot of future directions and not as much data, but now that COVID is getting beyond us, we can really, and now that we have a nice quorum, we can really start moving forward on the data set. But I really would just like to acknowledge the funding for this project, both from the Leon Levy Foundation, as well as a NARSAD grant to support this work. And then again, really would like to acknowledge Zainab, who's been with me for the past 3 years since I was a chief resident in research. And without her, I really couldn't have come this far in generating the data sets that we have. And again, would like to thank everyone that's assisted us, both from Paul Kenny's lab and the Russo lab, as well as Lee Shen and his group in helping us with the data analysis. And at this time, I'd like to open it up for questions.
Great. Thank you, Jess, for really exciting directional research you shared. So now we'd like to open to questions. You could either drop your question in the Q&A part, or you could also raise hands. I'm already seeing a question from Kavya.
Mm-hmm.
Do you want to ask question directly?
Sure, I can ask.
Kavya.
Oh, Kavya.
Hi, can you hear me? Okay, great, that worked. Thanks, Hiro, and Jess, great talk. This is so cool to see this coming together. I was really intrigued by the operant responding data. Mm-hmm. And I was just wondering if you had measured the glucose from the hyperglycemic and control mice when they were food restricted before the operant training, and whether, you know, because if, let's say, the hyperglycemic mice obviously have higher baseline glucose, and so the food restriction might alter that. And I was interested in how that interaction might be influencing the responding.
Yeah, we did measure their blood glucose before they went into the boxes, and the hyperglycemic mice were still hyperglycemic. In this particular cohort, they were quite hyperglycemic, with values in roughly the 350 to 450 range, even fasted. So they were much more hyperglycemic. For the controls, their values were roughly around 100, even when they were fasted. Because we're fasting them only to either 80 to 85% of their baseline body weight. So they are getting a little bit of food overnight, just to keep that weight maintained.
Okay, so they're still really different at that point. That makes sense.
Yes, they're still extremely different. And that's actually something that we will parse out more with the restaurant road task, is because in the restaurant road task, that's their only opportunity really to get food. So they're maintained on a fasted state, and the idea is that they have to learn to forage for all their food in a very limited time on that restaurant road task. And so we will be measuring their blood glucose both before and after each session in that restaurant row task to kind of get at that question.
Awesome, thank you.
Okay, great caveat. So I see a quick question from Nelly. So she's asking, what about hypoglycemia? Do you predict the contrasting result?
Yeah, that's a really good question. It's hard to say. So hypoglycemia is challenging because if there's a very narrow range in which an animal can be hypoglycemic and still functional. So I think it would be somewhat challenging to be able to assess that in these animals. Certainly we could do an insulin clamp and clamp them at different glycemic levels, but not within an operant box or within the restaurant row task. So it would be hard to say. Clinically, when patients are hypoglycemic, they do report more anxiety and have impaired cognition and processing speed. So I would actually expect their performance to be much worse and not be able to perform if we could get that to work in the operant boxes.
Yeah, I know it's a sort of an unfair question. Wonderful presentation, very interesting result, budding result.
Yes.
I wonder if, I think that it's really interesting in itself that the females were more resistant to becoming hyperglycemic.
Yes.
Is that correct?
It is correct.
So what do you make of that? I mean,
there's actually quite a bit of literature on it, and many other groups have demonstrated that fact. And it really does seem to be somewhat of an effect of estrogen, because if you overactivize the mice, then they become hyperglycemic, very similar to the males. So there must be some sort of downstream effect through insulin signaling or other alternative pathways to utilize glucose that allow these animals to compensate.
So estrogen might be a protective for a while.
Yes, and it is protective here. And I think largely this protective effect in the females is an evolutionary mechanism to preserve reproductive capability, reproductive status in females.
Yeah.
I mean, hyperglycemia is detrimental to the fetuses as well. So, there are a lot more mechanisms in play to keep it in a narrow range in the females.
Yeah, interesting.
Okay, so I see kind of a comment from Ms. Rita Williams. I'll read that. Not being a neuroscience person was very interesting. It would be nice if you could present the same program for dummies. I think that's a comment. Yep.
It is challenging to try and fit everything, all the background for both diabetes and reward processing together. So hopefully I managed to convey some of it to you, but definitely I know the habenula pathway is not the most well-known pathway in the reward circuitry.
Okay, so while I wait questions, maybe I ask one or two questions. So this is somewhat big picture level question, but when we think about hyperglycemia, are we only thinking about, is it primarily the insulin signaling we are focusing on? So then is the effect to habenula circuit you're focusing on, is that directly through insulin receptor expressed in this neuron or something indirect? Maybe you could elaborate the mechanistic aspect of the hyperglycemia itself.
Yes, so that's a very good question. So insulin is the only hormone that regulates glucose levels, but it does not. I mean, some of the effects are due to insulin signaling, but not exclusively. So some of the behavioral results can be reversed by replacing insulin or improving sensitivity of the insulin receptor, but not all of them, which then leaves the the possibility that it is the excess glucose levels themselves that are causing either oxidative damage or disrupting signaling within the neurons themselves. And so that is part of what we're trying to get at with this study first, is really looking at the effects of hyperglycemia. I think
Right.
to parse that out, to drill down more to the mechanism, I'm hoping that the sequencing will yield some pathways that we can then probe with either molecular or pharmacological tools in the setting of hyperglycemia, but also in normal animals, to really try and get down at that mechanism.
Yeah.
But I do agree. Part of it is signaling through the insulin receptor, but also secondary effects of just the excess blood glucose itself, which does cause damage to the structures.
So for the latter, it's the direct damage by the glucose itself. I see. So how about the insulin receptor? Are they expressing those habenula neural?
Yes, they
They
are.
are. Okay.
I
see. Is it too much to think about glucose sensor pathway?
We could look at it, and that's one thing that we would like to do is to see whether or not these neurons are responsive to glucose. Much of the work I did in Paul Kenny's lab was looking at the habenular neurons in nicotine addiction, and the intersection there that these neurons do cause some dysregulation of blood sugar in response to nicotine administration. And this does appear to be through the habenular neurons themselves.
I see.
So whether or not they are glucose responsive is still a question that's outstanding, but they do appear to have some direct effect on glucose regulation in the periphery.
I see. Thank you. Any questions? Okay, I have another question while I wait. So I have a question about the protocol. So you waited 6 weeks after the manipulation. Was there any rationale behind that? Did you explore waiting timing, or you have some literature that you end up deciding 6 weeks?
Yeah, so we wanted to wait a significant amount of time, but our choice of 6 weeks was somewhat arbitrary. We didn't want to look at it acutely because diabetes is a chronic illness, and hyperglycemia is persistent for quite some time. So we were interested really looking in more at the chronic effects, but most of the literature is examining the behavior in animals, has examined at least 30 days or more of hyperglycemia. So that was kind of why we decided to start with roughly that 5 to 6 weeks.
Right, I see. Somewhat relatively long enough period. Yeah. Given
also,
this is,
I didn't want the animals to be too old as well to have the factor of aging, although that's certainly part of diabetes as well.
Right. So the other direction could be starting from the younger animal, kind of getting to the critical period, especially if you have this two-hit idea. And I imagine type 1 diabetes may happen earlier in life. So
it does. So type 1 can actually happen at any age, even up into your 70s. You can acquire autoimmune diabetes. However, it is the most common autoimmune illness among children. And it is the more common type among children, with some poor children having onset very soon after birth, in fact. So yes, type 1 does affect more the children and would have more developmental implications. So yes, we can look at that as well.
Yeah, it's totally new field, so could be worth exploring the protocol for the hyperglycemia. Okay, let's see if others have questions.
Yeah, it looks like Rita was asking, were all mice approximately the same weight and have the same activity level? And so activity level we did measure, and they did have the same activity level. There was no difference in general locomotion between the two groups when we were assessing behavior or doing the operant task. However, the STZ treated mice are smaller than the control mice, and that is a well-known phenotype that's associated with diabetes. So I think when we were doing the operant task, the baseline weight for the control animals was around 30 to 35 grams, whereas for the STZ animals, it was around 25 grams. So they are significantly smaller than the control counterparts.
There's one more question.
Lailun Nahar asks, what is the connection between the function of glucose measured by blood and the brain behavior? And so that's a good question. So the brain is dependent on glucose for its function. And so it has a lot of mechanisms to ensure that adequate glucose is reaching the brain. So peripheral levels, blood levels of glucose are maintained in a certain range, largely to provide glucose supply to the brain. Hyperglycemia then puts peripheral glucose outside of this range. And there, as you can see, there are not really any mechanisms in place to control blood glucose in the absence of insulin or effective insulin signaling. So that's really kind of the whole gist of our research, is to see how does hyperglycemia affect brain behavior. A few studies have looked at injecting concentrated glucose directly into brain areas and found that hyperglycemia can impair synaptic transmission and can impair learning if injected directly into the hippocampus. But beyond that, you know, it's still quite an open box, despite diabetes being well known for 100 years now.
Okay. Maybe just a quick question. So do we need to think about damage to blood vessel itself after 6 weeks, potential blood-brain barrier damage, or that's not a factor for 6 weeks?
No, I think it is a valid concern. I think 6 weeks is perhaps not long enough. We're starting to examine that with some of our astrocyte staining, and we'll also do some endothelial markers to look at the integrity of the blood-brain barrier. But in patient populations where they looked at hyperglycemia and effects on mood, they did stratify them by vascular status. So those with retinopathy versus those who don't have retinopathy, which is either vascular damage or no vascular damage. And there was no difference between those two types of diabetic groups as far as mood went.
I see.
So I agree. I think it is a valid concern because certainly the neurovascular implications of diabetes are significant. But I think as well, it does not appear to be a critical factor in mood processing.
That's good to know. Okay, we just passed 1 p.m. So I think we had a great, exciting talk and discussion. So I'd like to thank Jess again for sharing your exciting talk, and thank you all for joining.
Yes,
Bye.
thank you very much.